Hi, this is Dr. David Greene, founder and CEO of R3 Stem Cell. Today the topic is actually an answer to a question. Is all amniotic fluid created equal for regenerative medicine therapy? And the answer is no, it's not. There's a lot of myths and misinformation that's been put out about amniotic fluid. Now, amniotic fluid is highly regulated by the FDA as a biologic, right? It's regulated as minimal manipulation, so you can't culture it or expand it or put significant enzymes or additives into it and turn it into a drug. However, in the FDA regulation of how it gets processed, there is some wiggle room so that, for instance, a lot of companies, when they manufacture it, they radiate it. And radiating it kills a lot of cells. It kills cytokines, it kills any kinds of stem cells and things like that. So that's pretty much a shortcut a lot of companies will take to get to the finish line before they cryogenically freeze it. But the FDA doesn't mandate what's called terminal sterilization. So if you don't radiate it, you can get a better amniotic product than one that is radiated. In addition, when the amniotic fluid is processed and then cryogenically frozen, first of all, it does not kill all the cells. Okay? That's been shown in studies over the last 20 years. You do need to add a preservative to the material to protect it while it's cryogenically frozen. If you add too much of the preservative, which is typically DMSO, then that in effect can kill all the biologic material. Any cells that were alive can be dead. So if you do what the lab we work with does and only add 3% DMSO, then it actually does what it's meant to do. It's meant to be a cryopreservative, and that's what it does. All right. Another thing that has, another myth that's been put out about amniotic is that when you freeze it, that it kills all the cells. Well, there was a study done about 10 years ago by a group led by a man named Betts, who showed that under 4 different freezing mechanisms, one very quickly, another one very slowly, and then 2 in between, that plenty of live cells were visible after thawing it out. Which makes sense, right? Because in a field such as fertility, when you have eggs being frozen and whatnot, that doesn't kill the egg, right? So it makes sense that cryopreservation, if done properly, doesn't kill the cells. So I just want you to keep in mind that when you have someone that pulls an amniotic product off the shelf and it's powder and they're adding saline to it to reconstitute it, that's not a real amniotic regenerative treatment, okay? A real one is a lab that uses only 3, 5%, less than 10% DMSO, that doesn't terminally sterilize the fluid, and that ends up with a product that has a lot of live components, which is what amniotic fluid is meant to be. Okay? Visit us online at r3stemcell.com. There's a lot of educational information on our site, as well as our YouTube channel, which has about 100 videos. And give us a call at 844-GET-STEM. We'd be happy to help get you in with a provider close to you to get your treatment. Thank you very much.